Earth emerges from the deep freeze of the Cryogenian period, a time when global glaciations turned the planet into a silent, white marble drifting through the void. This was not merely a pause in biological history, it was a crucible. The retreat of these massive ice sheets signaled the end of a planetary deadlock, triggering a rapid reconfiguration of the atmosphere, ocean chemistry, and crustal stability that would define the next 500 million years. When the ice finally relented, the rapid pulse of meltwater and the exposure of continental crust initiated a series of feedback loops that acted as an evolutionary catalyst. The release of trapped nutrients into the oceans created a surge in primary productivity, fueling the first complex life forms that would eventually navigate the complexities of multicellularity. This moment marks the transition from a world governed by static microbial dominance to one defined by dynamic, active, biological innovation. But the significance of this transition extends far beyond the emergence of complex organisms. It forces us to confront the vulnerability of planetary systems to atmospheric collapse. The mechanisms that allowed Earth to break free from the global ice trap represent a delicate, yet persistent resilience. If planetary homeostasis were slightly less robust, the Earth might have remained locked in a permanent sterilized state. A fate that potentially explains the absence of life on worlds that reached similar turning points, but lacked the geochemical triggers to restart. As we look deeper into this epoch, we encounter the fundamental tension between geological stability and biological progression. The evolution of the biosphere is inextricably linked to the tectonic and chemical evolution of the crust. The subsequent rise of oxygen levels and the formation of the ozone layer were not inevitable outcomes. They were consequences of specific, localized interactions between the lithosphere and the evolving life forms that began to reshape the very composition of the atmosphere. This interplay suggests that the history of life is not a linear march toward complexity, but a series of high-stakes, system-wide negotiations between the planet and its inhabitants. Understanding this process provides the necessary framework for assessing the viability of other worlds, as we begin to map the narrow conditions required to sustain the long-term trajectory of complex technological civilization. Emerging from the Cryogenian ice, the Ediacaran period reveals a world radically different from the microscopic reign of the preceding billions of years. We witness the first widespread appearance of macroscopic, multicellular life, a phenomenon often described as an evolutionary laboratory. These were not the ancestors of modern lineages in the conventional sense, but rather a collection of disparate, often bizarre body plans that defied the traditional categories of flora or fauna. Soft-bodied, frond-like, and disc-shaped organisms occupied the seafloor, lacking hard skeletons or shells, representing a unique experiment in biological architecture that operated under environmental pressures. We are only beginning to reconstruct. This period presents a critical data point in the history of life, the shift in atmospheric and oceanic chemistry. Evidence suggests a gradual rise in oxygen concentrations, a necessary precursor for the higher metabolic demands of multicellular organisms. However, this was not a linear increase. The oxygenation of the oceans was likely heterogeneous, creating localized niches where complex life could thrive, while other areas remained hypoxic. This environmental patchiness dictates the limits of our fossil record. We see a snapshot of success stories that managed to adapt to shifting chemical gradients, leaving behind soft tissue imprints in microbial mats that provide our only window into this alien-looking landscape. But the complexity of the Ediacaran goes beyond mere morphology. It is here that we find the first reliable evidence of biotic interaction that would set the stage for the arms race of subsequent eras. While traditional models once characterized the Ediacaran biota as a peaceful, passive community absorbing nutrients from the water column, recent trace fossil evidence suggests the onset of movement and, potentially, the earliest instances of predation or selective grazing. The emergence of burrowing organisms, though primitive, signifies a pivotal transition from the two-dimensional surface of the microbial mats to a three-dimensional utilization of the sediment. This move into the subsurface, often called the agronomic revolution, represents more than a shift in habitat. It changed the geochemistry of the seafloor forever. By disturbing the layers of microbial mats that had stabilized the substrate for eons, these organisms introduced oxygen deeper into the sediment and altered nutrient cycling. This is a profound example of biological engineering. Life was no longer merely responding to the environment, it was actively modifying the planet's surface. When we consider the Fermi Paradox, this phase becomes a crucial filter. The ability to move from passive existence to active environmental engineering is a prerequisite for any species capable of building a civilization. The Ediacaran illustrates that this transition is fraught with danger, as it requires overcoming the metabolic and structural limitations of single-celled life, while simultaneously surviving in a volatile, rapidly changing geochemical regime. We must also consider the limitations of our current interpretation. The lack of mineralization in these organisms means we are observing an incomplete phylogeny. Are these organisms the precursors to the Cambrian Explosion, or are they an evolutionary dead end that was pushed aside by more efficient, predatory competitors? The debate persists. Some models suggest that the Ediacaran biota was specialized for a high-nutrient, low-oxygen world, and as oxygen levels rose and competition intensified, their distinct biological strategy became untenable. This mirrors a recurring theme in the search for extraterrestrial life. The possibility that complex biology can flourish, only to be pruned by the very environmental changes it helps induce. As we analyze the decline of these forms, we are forced to reconcile with the fragility of early complexity. The transition from the Ediacaran to the Cambrian is not merely a change in the fossil record. It is a fundamental shift in the rules of survival. The development of hard parts and sophisticated sensory organs suggests that the era of experimentation was coming to a close, replaced by an era of intensive competition. If we view the history of life through the lens of a long-form timeline, the Ediacaran functions as the opening act, a period of immense creativity that defined the possibilities of multicellularity. Yet, it also serves as a warning. Ecological stability is an illusion maintained by the constant, often violent, negotiation between biology and the planet. This lesson remains relevant as we look toward the future of our own technological civilization, which now acts as the primary geological force on Earth, mirroring the primitive yet profound interventions of those first multicellular pioneers. Earth emerges from the deep freeze of the Cryogenian period, a time when global glaciations turned the planet into a silent, white marble drifting through the void. This was not merely a pause in biological history, it was a crucible. The retreat of these massive ice sheets signaled the end of a planetary deadlock, triggering a rapid reconfiguration of the atmosphere, ocean chemistry, and crustal stability that would define the next 500 million years. When the ice finally relented, the rapid pulse of meltwater and the exposure of continental crust initiated a series of feedback loops that acted as an evolutionary catalyst. The release of trapped nutrients into the oceans created a surge in primary productivity, fueling the first complex life forms that would eventually navigate the complexities of multicellularity. This moment marks the transition from a world governed by static microbial dominance to one defined by dynamic, active biological innovation. But the significance of this transition extends far beyond the emergence of complex organisms. It forces us to confront the vulnerability of planetary systems to atmospheric collapse. The mechanisms that allowed Earth to break free from the global ice trap represent a delicate, yet persistent resilience. If planetary homeostasis was slightly less robust, the Earth might have remained locked in a permanent sterilized state, a fate that potentially explains the absence of life on worlds that reached similar turning points but lacked the geochemical triggers to restart. As we look deeper into this epoch, we encounter the fundamental tension between geological stability and biological progression. The evolution of the biosphere is inextricably linked to the tectonic and chemical evolution of the crust. The subsequent rise of oxygen levels and the formation of the ozone layer were not inevitable outcomes. They were consequences of specific, localized interactions between the lithosphere and the evolving life forms that began to reshape the very composition of the atmosphere. This interplay suggests that the history of life is not a linear march toward complexity, but a series of high-stakes, system-wide negotiations between the planet and its inhabitants. Understanding this process provides the necessary framework for assessing the viability of other worlds, as we begin to map the narrow conditions required to sustain the long-term trajectory of complex technological civilization. Emerging from the cryogenian ice, the Ediacaran period reveals a world radically different from the microscopic reign of the preceding billions of years. We witness the first widespread appearance of macroscopic, multicellular life, a phenomenon often described as an evolutionary laboratory. These were not the ancestors of modern lineages in the conventional sense, but rather a collection of disparate, often bizarre body plans that defied the traditional categories of flora or fauna. Soft-bodied, frond-like, and disc-shaped organisms occupied the seafloor, lacking hard skeletons or shells, representing a unique experiment in biological architecture that operated under environmental pressures we are only beginning to reconstruct. This period presents a critical data point in the history of life: the shift in atmospheric and oceanic chemistry. Evidence suggests a gradual rise in oxygen concentrations, a necessary precursor for the higher metabolic demands of multicellular organisms. However, this was not a linear increase. The oxygenation of the oceans was likely heterogeneous, creating localized niches where complex life could thrive, while other areas remained hypoxic. This environmental patchiness dictates the limits of our fossil record. We see a snapshot of success stories that managed to adapt to shifting chemical gradients, leaving behind soft tissue imprints in microbial mats that provide our only window into this alien-looking landscape. But the complexity of the Ediacaran goes beyond mere morphology. It is here that we find the first reliable evidence of biotic interaction that would set the stage for the arms race of subsequent eras. While traditional models once characterized the Ediacaran biota as a peaceful, passive community absorbing nutrients from the water column, recent trace fossil evidence suggests the onset of movement and, potentially, the earliest instances of predation or selective grazing. The emergence of burrowing organisms, though primitive, signifies a pivotal transition from the two-dimensional surface of the microbial mats to a three-dimensional utilization of the sediment. This move into the subsurface, often called the agronomic revolution, represents more than a shift in habitat. It changed the geochemistry of the seafloor forever. By disturbing the layers of microbial mats that had stabilized the substrate for eons, these organisms introduced oxygen deeper into the sediment and altered nutrient cycling. This is a profound example of biological engineering. Life was no longer merely responding to the environment, it was actively modifying the planet's surface. When we consider the Fermi Paradox, this phase becomes a crucial filter. The ability to move from passive existence to active environmental engineering is a prerequisite for any species capable of building a civilization. The Ediacaran illustrates that this transition is fraught with danger, as it requires overcoming the metabolic and structural limitations of single-celled life, while simultaneously surviving in a volatile, rapidly changing geochemical regime. We must also consider the limitations of our current interpretation. The lack of mineralization in these organisms means we are observing an incomplete phylogeny. Are these organisms the precursors to the Cambrian Explosion, or are they an evolutionary dead end that was pushed aside by more efficient, predatory competitors? The debate persists. Some models suggest that the Ediacaran biota were specialized for a high-nutrient, low-oxygen world, and as oxygen levels rose and competition intensified, their distinct biological strategy became untenable. This mirrors a recurring theme in the search for extraterrestrial life: the possibility that complex biology can flourish, only to be pruned by the very environmental changes it helps induce. As we analyze the decline of these forms, we are forced to reconcile with the fragility of early complexity. The transition from the Ediacaran to the Cambrian is not merely a change in the fossil record, it is a fundamental shift in the rules of survival. The development of hard parts and sophisticated sensory organs suggests that the era of experimentation was coming to a close, replaced by an era of intensive competition. If we view the history of life through the lens of a long-form timeline, the Ediacaran functions as the opening act, a period of immense creativity that defined the possibilities of multicellularity. Yet, it also serves as a warning. Ecological stability is an illusion maintained by the constant, often violent, negotiation between biology and the planet. This lesson remains relevant as we look toward the future of our own technological civilization, which now acts as the primary geological force on Earth, mirroring the primitive yet profound interventions of those first multicellular pioneers. For tens of millions of years, Earth was a sealed system, a pristine, white sphere drifting through the vacuum, locked in a planetary glaciation known as the Cryogenian period. The albedo effect, where brilliant ice reflects solar radiation back into space, had trapped the climate in a self-sustaining feedback loop of extreme cold. Beneath kilometers of solid ice, the biological clock was practically stalled, restricted to microscopic survival in isolated geothermal oases. But geological mechanics operate on a scale that ignores biological stagnation. Deep beneath the frozen crust, tectonic volcanism continued. Without liquid oceans exposed to wash carbon dioxide from the atmosphere through silicate weathering, the greenhouse gas steadily accumulated. Over millions of years, the atmosphere transformed into a pressurized thermal blanket. When the ice finally reached its melting threshold, the collapse of the global glaciation was violently rapid. The retreating glaciers ground continental rock into fine dust, flushing millions of years of trapped phosphorus and iron directly into the barren oceans. This sudden chemical shockwave fundamentally altered marine chemistry, driving a massive spike in atmospheric oxygen. And right here, this geological mechanism connects to a much larger cosmological question. Earth's survival through this deep freeze was not mathematically guaranteed. In the study of astrobiology and the Fermi Paradox, a permanent global ice state is considered a highly probable great filter. A rocky planet slightly less volcanically active or orbiting just a fraction further from its host star would never thaw. It would remain a sterile, frozen monument. Earth narrowly crossed this atmospheric threshold. The resulting nutrient-rich, highly oxygenated oceans became the ultimate biological crucible. The 3 billion year stagnation of single-celled dominance was shattered, forcing biological systems to adapt to a radically volatile new chemistry and triggering the great evolutionary machinery that would soon build the first complex, multicellular architectures. Out of the chemical chaos of the melting planet, the first visible structures of complex life begin to emerge in the fossil record. This period, known as the Ediacaran, introduces a biological paradigm so utterly alien that paleontologists debated for decades whether these organisms were animals, fungi, or a completely extinct kingdom of life. We do not see the familiar bilateral symmetry of modern fish or arthropods. Instead, the shallow sunlit seas were populated by macroscopic, soft-bodied organisms that utilized a fractal architecture. Creatures known as rangeomorphs grew by endlessly repeating the same branching geometric patterns, expanding their surface area to extreme proportions. They possessed no mouths, no digestive tracts, and no central nervous systems. They simply anchored themselves to the microbial mats that carpeted the sea floor and absorbed dissolved carbon and nutrients directly from the water column through osmosis. This represents a completely divergent operating system for biology. In the context of astrobiology, the Ediacaran biome serves as a vital template. If we ever detect complex life in the subsurface oceans of moons like Europa or Enceladus, where sunlight is absent and chemical gradients dictate survival, it is highly probable that such life will favor this exact osmotic, high surface area architecture over the active, energy-intensive body plans we see on modern Earth. But this passive, garden-like ecosystem was constrained by a severe physical limitation. Relying on diffusion for energy means an organism can only grow as large as the chemical gradient allows before its internal volume outpaces its absorbing surface area. To break this mathematical limit, life had to invent a new mechanism: motility. The fossil record shifts from preserving just body impressions to preserving trace fossils, the tracks, trails, and burrows of organisms actively moving across and through the sediment. We see the faint, rhythmic, crawling marks of creatures like Dickinsonia, sweeping across the bacterial mats to digest them from below. This transition from static existence to directional movement required a massive increase in metabolic energy, demanding higher localized oxygen concentrations. More importantly, directional movement requires spatial awareness. The organism must be able to differentiate between a nutrient-rich direction and a barren one. At a planetary scale, the implication of this shift changes entirely. The moment life began to move with purpose, the foundation was laid for sensory organs, and eventually, the central nervous systems required to process that environmental data. Without this specific metabolic leap, biological complexity would have permanently flatlined at the level of fractal sponges, rendering the evolution of technological intelligence impossible. However, the invention of movement introduced a far darker, infinitely more powerful evolutionary driver. Deep in the Ediacaran strata, researchers discovered fossils of tiny cone-shaped organisms called Claudina, representing some of the earliest known creatures to build hard, calcified shells. For a long time, the sudden appearance of armor was a mystery, until high-resolution imaging revealed perfectly circular, microscopic boreholes drilled directly through the sides of these shells. This is not a geological artifact. It is the definitive signature of a predator, something active, equipped with rudimentary chemical or mechanical weaponry, was hunting Claudina. This single, microscopic hole in a primitive shell represents the initiation of the biological arms race. the rules of survival on Earth were rewritten overnight. Natural selection ceased to be merely a game of surviving the environment and became a ruthless competition of surviving other biological entities. This development leads to a much deeper planetary consequence. The evolutionary pressure to hunt and to avoid being hunted forced life to optimize. It drove the development of visual receptors, faster muscular responses, and the neurobiology required to predict a target's movement. Intelligence, in its most foundational form, was forged not as an inevitable peak of evolution, but as a survival mechanism in a suddenly hostile, predatory ocean. And right here, this biological shift begins to connect with the question of civilizational longevity and the Great Filter. The end of the Ediacaran period is marked by a massive sudden extinction event, wiping out nearly all the fractal, soft-bodied pioneers. The prevailing scientific model suggests they were not killed by a meteor or a sudden freeze, but by the very ecosystem they helped create. As new, mobile organisms evolved to burrow deep into the seafloor to hide from predators or seek new nutrients, they systematically destroyed the stabilizing microbial mats that the Ediacaran organisms relied upon. This event, known as the Cambrian substrate revolution, is a profound example of biological engineering causing catastrophic environmental collapse. The biosphere inadvertently destroyed its own foundation. When we look at the Fermi Paradox and wonder why we see no evidence of advanced civilizations in the galaxy, the Ediacaran extinction provides a chilling, localized model. It demonstrates that as soon as a biological system gains the capacity to actively engineer its environment, whether through primitive burrowing or advanced industrial technology, it immediately faces the existential risk of destabilizing the delicate geological and chemical systems that allowed it to emerge in the first place. The destruction of the Ediacaran microbial mats was not an end, but a clearing of the board. As the Cambrian period begins, approximately 538 million years ago, the fossil record undergoes a transformation so abrupt and profound that it has troubled evolutionary biologists since Darwin. This is the Cambrian Explosion, but the term itself is slightly misleading. It was not a sudden, spontaneous generation of life from nothing. Rather, it was a sudden explosion of biological visibility. For the first time in Earth's history, living organisms began to systematically extract dissolved minerals from the surrounding ocean to construct hard, three-dimensional architectures. This process, known as biomineralization, represents a fundamental structural paradigm shift. Organisms began utilizing calcium carbonate and calcium phosphate to build shells, spines, and ultimately, internal skeletons. The immediate assumption is that this biological armor was a direct defensive response to the predatory pressures that began in the late Ediacaran. While protection was undoubtedly a crucial factor, biomineralization solved a much deeper mechanical problem. Soft tissue cannot exert significant force without a rigid anchor. By building hard parts, life discovered mechanical leverage. Muscles could now attach to rigid nodes, allowing for rapid contraction, articulated movement, and the sheer physical power required to swim against currents or crush the defenses of competing organisms. But constructing and carrying mineralized armor requires an immense biological budget. It demands a metabolic output far exceeding the capabilities of passive, diffusion-based organisms. For decades, the dominant scientific model proposed that a sudden massive spike in global atmospheric oxygen was the singular trigger that unlocked this biological revolution. Oxygen is the ultimate biological fuel, enabling highly efficient aerobic respiration to power complex nervous systems and heavy, mineralized bodies. However, modern geochemical analysis has profoundly complicated this clean narrative. When geologists examine the trace metal concentrations in early Cambrian black shales, specifically isotopes of molybdenum and uranium, which precipitate differently depending on the availability of oxygen, they do not find evidence of a uniformly highly oxygenated ocean. Instead, the data reveals a volatile, fluctuating chemical environment. The deep oceans remained largely anoxic, hostile to complex life, while oxygen levels in the shallow continental shelves repeatedly spiked and crashed. This leads to a much deeper scientific consequence. If global oxygen levels were dangerously unstable, the Cambrian explosion did not occur in a stable, optimized paradise, but rather in isolated, high-stakes incubators. We call these localized zones oxygen oases. In these shallow, sunlit waters, photosynthetic algae generated localized pockets of breathable water, surrounded by vast stretches of lethal, low-oxygen ocean. This spatial restriction forced populations into intense, localized competition, accelerating the evolutionary arms race within these confined ecological arenas. At a planetary scale, the implication of the oxygen oasis model changes entirely how we view the emergence of complexity. It suggests that a planet does not need a globally perfect atmosphere to ignite an evolutionary explosion. It only requires highly active, localized chemical gradients. This concept is revolutionary for astrobiology and the search for extraterrestrial intelligence. When we analyze the atmospheric spectra of distant exoplanets, a low overall oxygen signature might not indicate a dead world. It could be masking a planet functioning exactly like early Cambrian Earth, harboring fierce, highly advanced ecosystems in isolated geochemical pockets. And right here, this geological threshold connects directly to the concept of the Great Filter. If complex life requires a strict, globally uniform oxygen threshold to evolve, then the universe might be largely barren, as stable, high-oxygen atmospheres are difficult to maintain over geological timeframes. But if complex life is forged in the volatile, fluctuating margins, surviving chemical crashes, and taking refuge in temporary oases, then the evolutionary filter is not about planetary perfection, but about biological resilience. This relentless pressure in the Cambrian oases drove the next critical innovation: advanced sensory processing. As predator and prey engaged in this hyper-accelerated arms race, simply having a hard shell was no longer sufficient. Survival required the ability to detect threats before they made physical contact. This ecological demand forced the evolution of the first true visual systems. Organisms, like the early trilobites, began to biomineralize calcite into precisely aligned crystalline lenses, creating compound eyes capable of resolving distinct shapes and movements in the water column. This was not just an anatomical upgrade. It was the birth of active information processing. The ocean ceased to be a dark, chemical void and became a defined spatial theater. The influx of visual data demanded a corresponding leap in neurological processing power, laying the unalterable foundation for the centralized brains that would eventually map the cosmos. If this precise sequence of geochemical volatility, localized competition, and biomineralization had not occurred, the evolutionary trajectory would have remained blind, and the concept of an observer within the universe might never have materialized. The visual and biomechanical revolutions of the early Cambrian did not merely create a defensive arms race. They engineered the Earth's first true biological hierarchy. As the shallow seas filled with armored, highly mobile organisms, a massive amount of metabolic energy became locked within complex biological structures. This high-density energy source inevitably drove the evolution of the first apex predators, radically shifting the flow of planetary biomass. At the absolute top of this new food web sat Anomalocaris, a free-swimming predator that reached lengths of over a meter, a true leviathan in a world where most life was measured in centimeters. It possessed large stalked compound eyes, equipped with thousands of individual lenses, providing extraordinary visual acuity to track movement across the sunlit water column. Its defining feature, however, was a pair of grasping, articulated appendages designed to capture and crush the calcified shells of its prey, funneling them into a square, serrated mouth. The existence of such a highly specialized predator indicates that the oceanic ecosystem had achieved a profound level of energetic efficiency. Trophic levels were now firmly established, meaning energy captured by photosynthesis at the microscopic level was successfully being transferred up through multiple layers of consumption. At a planetary scale, the meaning of this changes entirely. When astrobiologists consider the potential for complex life in subsurface alien oceans, the emergence of a trophic apex is considered a definitive marker of a mature biosphere. Without apex predators regulating populations, prey species would exhaust their local resources and collapse. Predation acts as the ultimate stabilizing force, an aggressive mechanism of planetary homeostasis that forces continuous systemic adaptation. But the biological hierarchy of the Cambrian oceans leads to a much deeper consequence. While the heavily armored arthropods dominated the physical landscape, their strategy contained a fatal architectural flaw. An exoskeleton provides excellent immediate protection, but it severely restricts physical growth and requires a highly vulnerable molting process. Furthermore, it limits the internal space available for a centralized nervous system to expand. And right here, this structural limitation connects to the most critical, yet visually unimpressive, biological development of the era. Hidden within the fossil beds of the Burgess Shale are the remains of a creature known as Pikaia. It possessed no protective shell, no compound eyes, and no grasping claws. It was essentially a small, swimming ribbon of muscle, desperately trying to survive in a sea of heavily armed predators. However, running down the dorsal length of Pikaia was a stiff, flexible rod of cartilage known as a notochord. This is the first known iteration of an internal skeleton. This internal scaffolding was a revolutionary piece of biological engineering. Instead of enclosing the body in heavy stone, the organism built a lightweight core that muscles could anchor against, allowing for rapid, undulating movement. More importantly, this structure provided a secure, dedicated conduit for a dorsal nerve cord. This was the foundational architecture for the vertebrate nervous system. The notochord allowed for continuous physical growth without molting and laid the groundwork for a heavily protected, centralized brain casing that would evolve hundreds of millions of years later. If this precise anatomical configuration had not appeared, or if the fragile chordate lineage had been hunted to extinction by the dominant radiodont predators, the evolutionary trajectory toward human intelligence would have been permanently erased. This realization introduces a profound tension into our understanding of the Great Filter. The survival of the vertebrate blueprint was not a guaranteed outcome of the Cambrian Explosion. It was a statistical anomaly. The lineage that would eventually construct radio telescopes and split the atom survived its earlier stages as a marginalized, soft-bodied scavenger, constantly evading the apex predators of its day. This suggests that the path to a technological civilization is incredibly narrow. The Great Filter might not always be a dramatic cosmic catastrophe like a supernova or an asteroid impact. Instead, the filter could simply be the overwhelming probability that the specific, vulnerable, biological architectures required for high-level intelligence, are quietly consumed in the hyper-competitive oceans of a young world, long before they ever get the chance to leave the water. The fundamental body plans of complex life were drafted during the Cambrian, but the subsequent Ordovician period was the era of mass production and hyper-specialization. Beginning roughly 485 million years ago, the oceans underwent a transformation known as the Great Ordovician Biodiversification Event. If the Cambrian Explosion was the sudden appearance of isolated evolutionary prototypes, the Ordovician was the systematic colonization of every available ecological niche. Within a few tens of millions of years, the diversity of marine life tripled. Families and genera multiplied exponentially without introducing entirely new phyla, representing a massive optimization of the existing biological software. This staggering explosion of diversity was not driven by a sudden genetic mutation, but by a profound shift in planetary geology. During the Ordovician, intense tectonic activity caused the continental plates to drift apart, creating the largest expanse of shallow tropical continental shelf seas in Earth's history. These submerged platforms provided the perfect incubation zones. Sunlight could penetrate all the way to the seafloor, supercharging photosynthetic algae and establishing a massive, stable, energetic baseline for the entire food web. In these warm, sunlit shallows, life initiated its first large-scale architectural projects. We see the emergence of the first true biological megastructures, primitive coral reefs. Early reef builders, including tabulate and rugose corals, alongside calcium-secreting sponges known as stromatoporoids, began to extract heavy minerals from the ocean to construct sprawling, rigid frameworks across the sea floor. This represents a monumental shift in planetary engineering. A flat, sandy seafloor has a mathematically limited surface area. A reef, however, is a biological fractal. It creates three-dimensional topography, generating thousands of microhabitats, shifting local ocean currents, and providing shelter from the increasingly sophisticated pelagic predators navigating the waters above. At a planetary scale, the significance of this reef building alters our fundamental understanding of habitability. When we search for astrobiological targets, the presence of liquid water is often considered the ultimate metric for a living world. But the Ordovician teaches us that a global ocean is merely a blank canvas. It is the active geology of a planet that forces life to innovate. If a water-rich exoplanet possesses a stagnant lid, crust lacking plate tectonics, it will never form these crucial, shallow, continental shelves. Without the isolating and dynamic environments of shifting tectonic plates, life on such a world might remain permanently suspended in a state of low diversity, microbial simplicity. The engine of evolution is not just water and carbon. It is the restless, fracturing machinery of the planetary crust itself. But the issue does not stop there. The ecological specialization occurring within these Ordovician reefs created a vertical expansion of the food web. For the first time, dense populations of filter feeders emerged, capturing floating plankton and transferring immense amounts of organic matter from the surface down to the benthic zones. Meanwhile, early cephalopods evolved highly advanced buoyancy control, utilizing gas-filled chambers in their shells to detach from the seafloor entirely and dominate the open water column. The ocean became a fully occupied, three-dimensional matrix of biological energy transfer. This leads to a much deeper consequence regarding the stability of living worlds. The sheer efficiency of the Ordovician biosphere introduced a severe hidden vulnerability. To build millions of square kilometers of reefs and countless billions of heavily armored shells, marine life extracted unfathomable quantities of calcium and carbonate directly from the water. Simultaneously, the exposed tectonic landmasses underwent intense silicate weathering, a chemical reaction that physically pulls carbon dioxide out of the atmosphere and washes it into the sea. The biosphere and the geosphere were working in tandem, acting as a colossal runaway carbon sink. And right here, this geological mechanism begins to connect with the larger question of civilizational and biological longevity. The relentless biological expansion was actively terraforming the planet, rapidly stripping away its thermal greenhouse blanket. The very success of the Ordovician and biosphere, its staggering diversity, its mastery of biomineralization, and its complete colonization of the continental shelves was pushing the planetary climate toward a catastrophic tipping point. If this mechanism holds true across the cosmos, it suggests a terrifying phase in the life cycle of any complex biosphere, serving as a localized model for the Great Filter. A highly efficient, expanding biological system can alter its host planet's atmospheric chemistry so rapidly that it triggers its own eradication. The hyperactivity of the Ordovician was not a state of eternal biological harmony. It was an unsustainable metabolic peak. By aggressively optimizing for survival in the shallow seas, life inadvertently initiated a global atmospheric drawdown, setting the stage for a dramatic drop in temperature that would soon plunge the Earth into a lethal, deep freeze extinction, proving that on a geological timescale, extreme biological success is often the immediate precursor to planetary scale collapse. The Ordovician biosphere had engineered a staggering biological matrix across the shallow continental shelves. But this extreme optimization masked a profound vulnerability. Approximately 443 million years ago, this thriving planetary ecosystem collapsed in a catastrophic event that wiped out nearly 85% of all marine species. In the geological strata, the die-off is abrupt, marking the first of the Earth's Big Five mass extinctions. While traditional models place the blame solely on a sudden, severe global ice age, the precise trigger for such an immediate and extreme climatic shift has driven modern science to look beyond the Earth itself. This search for a trigger introduces one of the most terrifying hypotheses in astrobiology: a direct hit from a gamma-ray burst. A gamma-ray burst, or GRB, is the most luminous electromagnetic event occurring in the universe, typically generated by the collapse of a massive star into a black hole or the collision of neutron stars. If a GRB occurred within a few thousand light years of Earth and its energetic beam was aimed directly at our planetary system, the initial strike would be invisible. The intense pulse of gamma radiation would flash over the Earth in a matter of seconds, perhaps minutes. It would not physically incinerate the surface, but its interaction with the upper atmosphere would be devastating. The high-energy photons would shatter molecular nitrogen and oxygen, forcing them to recombine into toxic nitrogen dioxide gases. This sudden chemical restructuring would instantly strip away the planet's protective ozone layer, bathing the Earth's sunlit hemisphere in lethal levels of ultraviolet radiation from our own sun. But the issue does not stop there. The formation of a dense nitrogen dioxide smog would act as an atmospheric shield, blocking visible sunlight from reaching the surface. This creates a terrifying dual effect: extreme ultraviolet radiation poisoning for any organisms in the shallow surface waters, coupled with a rapid global temperature drop as solar heat is reflected back into space. This theoretical cosmic trigger perfectly aligns with the subsequent geological evidence. The sudden formation of massive ice sheets over the supercontinent of Gondwana which was situated at the South Pole. At a planetary scale, the implication of the GRB hypothesis changes our entire framework for planetary habitability. If this model is accurate, it introduces a hard cosmic limit to the Great Filter. It implies that planetary stability is an illusion. A civilization could evolve on a geologically perfect world, master its own climate, and achieve global peace, only to be sterilized without warning by the death throes of a star completely outside its control. This suggests that the galaxy itself possesses a habitable zone, an orbital region far enough from the dense, volatile galactic core to minimize the statistical probability of being caught in the crossfire of high-energy cosmic events. Earth's survival may simply be a byproduct of its location in the quiet suburbs of the Milky Way. Whether initiated by a cosmic blast or purely internal tectonic cycles, the resulting glaciation drained the oceans. As water was locked away in immense polar ice caps, global sea levels plummeted by over 100 meters. The vast, shallow continental shelves, the incubation zones of Ordovician complexity, were suddenly exposed to the dry, freezing air. The massive reef networks collapsed, and the highly specialized food webs disintegrated. Yet, from this near sterilization, the subsequent Silurian period reveals a profound evolutionary pivot. The oceans had proven to be a volatile and deadly trap. For millions of years, the barren rocky continents had been bombarded by radiation and extreme weather, devoid of anything more complex than bacterial crusts. But the intense evolutionary pressure of the extinction forced a radical adaptation. The ultimate escape was onto the land. Here, we witnessed the emergence of the first vascular plants, such as the rudimentary Cooksonia. To colonize dry rock, these organisms had to solve two monumental engineering problems: defying gravity and surviving desiccation. They achieved this by evolving a revolutionary internal plumbing system known as vascular tissue. Microscopic tubes of xylem and phloem allowed them to pull water and dissolve minerals upward from the soil and distribute sugars generated by photosynthesis downward. To keep these tubes from collapsing under their own weight or the force of the wind, life synthesized lignin, a highly complex polymer that provided rigid structural support. This leads to a much deeper consequence for the Earth's long-term future. The invasion of the continents by vascular plants was not just a biological expansion. It was an aggressive, planetary-scale terraforming project. As primitive roots probed the sterile rock, they secreted organic acids that dissolved the stone, physically breaking down the Earth's crust to create the very first true soils. This dramatically accelerated the rate of silicate weathering, once again drawing carbon dioxide out of the atmosphere and establishing a new, slower, and more stable climatic thermostat. If we look closely at this Silurian transition, we realize it is the absolute prerequisite for our current existence. The creation of soil and the stabilization of the terrestrial environment paved the way for arthropods and eventually vertebrates to leave the water. More profoundly, the invention of lignin created a biological material so robust that for millions of years, almost nothing could decompose it. These early plants would eventually accumulate, bury themselves, and compress under geological heat to form the vast subterranean energy reserves of coal. If this specific vascular leap had not occurred, the Earth's surface would have remained a barren rock, and the primary energy source that fueled the Industrial Revolution, the critical threshold of our modern technological civilization, would never have existed. While the invasion of the land during the Silurian set the stage for terrestrial ecosystems, the absolute peak of biological engineering was simultaneously unfolding beneath the waves. As we enter the early Devonian period, roughly 490 million years ago, the marine environment recovered from the Ordovician freeze. and transformed into a high-density theatre of war. This era is widely known as the Age of Fishes, but from a purely mechanical perspective, it should be recognized as the era of the first true biological weapons. For billions of years, the consumption of other organisms was restricted by the physical size of an animal's mouth. Early vertebrates were jawless, relying on passive filter feeding or weak suction to consume soft-bodied prey or organic debris. They were essentially vacuum cleaners scavenging the seafloor. But in the early Devonian, a radical architectural mutation occurred within the vertebrate skeletal system. The anterior cartilaginous arches that supported the gills, structures originally dedicated exclusively to respiration, began to migrate forward. They enlarged, developed a hinge, and fused with the skull. This is the invention of the vertebrate jaw. It stands as one of the most violent and consequential biomechanical leaps in planetary history. With a hinged jaw, consumption was no longer limited to swallowing prey whole. Animals could now bite, tear, and mechanically dismantle biological matter that was significantly larger than themselves. At a planetary scale, the meaning of this changes entirely. A jaw is not just a weapon, it is an advanced energy extraction mechanism. It allowed predators to crack open the heavily armored shells of Cambrian and Ordovician survivors, accessing the high-density, calorie-rich proteins and fats hidden inside. In the context of astrobiology and the potential for extraterrestrial intelligence, the evolution of a mechanical mastication system represents a critical threshold. High-level intelligence requires a centralized nervous system, and a centralized brain is an absolute metabolic furnace. It demands a massive, continuous caloric budget. If a biosphere fails to invent a mechanism like the jaw to rapidly process and extract dense energy from its environment, evolutionary complexity will permanently flatline. The alien oceans of Europa or Enceladus might harbor life, but without the specific biomechanical leap of the hinged jaw, that life is highly unlikely to possess the metabolic fuel required to evolve into a technological civilization. The sudden introduction of the jaw triggered an unprecedented arms race in the Devonian seas. We see the rapid diversification of the placoderms, a class of terrifyingly modified armored fish. Placoderms did not merely have scales. They encased their heads and upper torsos in thick, interlocking plates of solid bone. Their jaws were fundamentally different from modern teeth. Instead of individual enamel spikes, they utilized the massive edges of their skull plates, which constantly ground against each other to maintain a razor-sharp, self-sharpening edge. They functioned like industrial hydraulic shears, capable of snapping the spine of any competing vertebrate. Fossil evidence from this period reveals deep gouges and shattered armor plates, painting a picture of an ocean where extreme violence was the baseline of existence. But the problem does not stop at raw physical power. Heavy armor and crushing bite forces require an immense physiological toll. A heavily armored placoderm is a biological tank, meaning it is slow, requires vast amounts of food to maintain its bone density, and has terrible maneuverability in open water. This leads to a much deeper consequence in the evolutionary timeline. While the placoderms dominated the immediate hierarchy through sheer force, two marginal, less impressive lineages were quietly investing in a completely different survival strategy: sensory dominance and mobility. Deep in the Devonian strata, we find the first true cartilaginous fish, the ancestors of modern sharks. Instead of heavy bone, they built skeletons of lightweight cartilage, sacrificing invulnerability for speed. More importantly, they began to develop the ampullae of Lorenzini, a network of specialized pores capable of detecting the faint electrical fields generated by the muscle contractions of hidden prey. They stopped trying to out-armor their opponents and instead evolved the ability to perceive the invisible electromagnetic matrix of the ocean. Simultaneously, the lobe-finned fish, the Sarcopterygians, developed thick, muscular appendages supported by a robust, internal bone structure. While placoderms were trapped in the heavy competitive zones of the open reefs, the lobe-finned fish used their specialized fins to navigate the shallow, oxygen-poor coastal swamps where the heavy dreadnoughts could not follow. And right here, this divergence connects to our very existence. The impregnable armor of the placoderms proved to be an evolutionary dead end, leading to their total extinction by the end of the Devonian. The universe, it seems, favors adaptability over static defense. The seemingly vulnerable, fleshy fins of the Sarcopterygians contained a very specific internal geometry, one stout bone attached to two smaller bones, branching into a complex array of radial bones. This exact sequence was the unalterable blueprint for the femur, the tibia, the fibula, and the human hand. The intense, weaponized pressure of the Devonian oceans forced these early vertebrates to the margins, inadvertently equipping them with the exact mechanical architecture necessary to eventually crawl out of the water, manipulate tools, and point telescopes at the stars. The invasion of the land was not merely a biological milestone, it was a fundamental rewiring of the Earth's geochemical engine. During the latter half of the Devonian period, terrestrial ecosystems underwent a massive vertical and horizontal expansion. The evolution of the seed-freed plants from the immediate vicinity of water, while the development of extensive, deep-penetrating root systems allowed them to anchor into the harsh continental interior. This led to the rise of the first true forests, dominated by colossal, fern-like trees such as Archaeopteris, which formed sprawling canopies across the supercontinents. These deep roots initiated a planetary scale mining operation. As they aggressively pushed through the upper crust, they secreted organic acids that shattered the underlying silicate bedrock, creating the first deep, stable soils in Earth's history. This biological weathering was an incredibly efficient terraforming mechanism, physically extracting essential minerals like phosphorus and iron from the stone. But matter on a planetary scale is never simply created or destroyed. It is displaced. The immense volume of nutrients liberated by these roots did not remain locked in the terrestrial ecosystem. Washed away by rainfall and newly formed river networks, millions of tons of biological fertilizer were continuously flushed directly into the shallow, sunlit epicontinental seas. This influx of raw chemical fuel triggered a catastrophic biological reaction. Microscopic marine algae, suddenly bathed in a limitless food supply, multiplied exponentially. The fossil record indicates the formation of massive global algal blooms that choke the surface waters. However, this hyperproductivity is a death sentence for a marine ecosystem. When these unfathomable quantities of algae completed their short life cycles and sank into the depths, the subsequent bacterial decomposition process consumed practically every available molecule of dissolved oxygen in the water column. This leads to one of the most terrifying ecological paradoxes in the geological record. The vast, vibrant oceans of the Devonian were not sterilized by a sudden cosmic impact or an immediate burst of radiation. They were slowly suffocated by the rampant evolutionary success of the terrestrial biosphere. The forests effectively asphyxiated the seas. This phenomenon, known as oceanic anoxia, turned the vibrant reef systems into sprawling, toxic dead zones. The highly specialized, heavily armored placoderms, requiring immense amounts of oxygen to sustain their massive metabolic engines, were completely eradicated. The apex predators of the age were defeated by the quiet growth of trees thousands of miles away. In the context of astrobiology and the search for extraterrestrial intelligence, this mechanism introduces a disturbing variant of the Great Filter. It suggests that a planetary biosphere does not require a technologically advanced civilization to trigger a global climate collapse. Biological evolution alone, optimizing purely for its own immediate survival and expansion, is entirely capable of engineering its own mass extinction through unchecked geochemical efficiency. If an alien world develops a highly aggressive terrestrial ecology, it might inherently destabilize its oceanic foundation, resetting the evolutionary clock long before any species develops the cognitive capacity to understand what is happening. But the complexity of the late Devonian extinction events does not end with forest growth. While the eutrophication model explains the widespread marine anoxia, isotopic analysis of the rock strata reveals sudden spikes in mercury and heavy metals, signatures that cannot be produced by rotting algae. This points to a concurrent geological nightmare: the eruption of massive volcanic provinces, likely the Viliwi traps in modern-day Siberia. This introduces a dual-strike scenario that highlights the extreme fragility of complex life. The biological terraforming of the continents severely compromised the oceanic immune system, leaving the global biosphere entirely defenseless when deep mantle tectonics unleashed a wave of toxic volcanic greenhouse gases. And right here, this catastrophic convergence defines our exact evolutionary lineage. The catastrophic drop in oceanic oxygen meant that survival in the water required a radical new architecture. The marginalized lobe-finned fish, already living in the shallow, oxygen-poor coastal swamps, possessed a primitive lung, initially evolved merely to gulp surface air during dry spells. In an anoxic ocean, this rudimentary organ became the ultimate survival mechanism. The Devonian extinction proves that the traits necessary to build a technological civilization, such as air-breathing lungs and articulated limbs, were not forged in an era of biological prosperity. They were refined in a suffocating, dying ocean, driven by the desperate biological imperative to escape a collapsing ecosystem. If the Devonian forests had not triggered this planetary asphyxiation, our ancestors might have remained comfortably in the water, and the evolutionary pressure to conquer the dry land would never have materialized. The escape from the dying Devonian oceans brought vertebrate life into a radically transformed terrestrial environment, initiating the Carboniferous period, roughly 359 million years ago. But the true architect of this new era was not animal life. It was the sprawling, global rainforests. These primitive vascular plants had perfected the synthesis of lignin, the complex structural polymer that allowed them to build massive wooden trunks and reach unprecedented heights. However, this biological innovation introduced a profound mechanical flaw into the Earth's recycling system. While the forests were manufacturing millions of tons of dense wood, the biological countermeasures, the specific fungi and bacteria capable of degrading and decomposing lignin, had not yet evolved. This creates a localized geological anomaly that defies the normal operational parameters of a stable biosphere. When these colossal trees died, they did not rot. They collapsed into vast, stagnant swamp networks, piling up layer after layer, essentially permanently removing gigatons of carbon from the active biological cycle. Because photosynthesis extracts carbon dioxide and releases oxygen, this massive carbon burial broke the atmospheric equilibrium. Without the decomposing organisms to consume that oxygen and release carbon dioxide back into the air, the atmospheric equation wildly destabilized. global oxygen levels skyrocketed from the current 21% to a staggering 35%. At a planetary scale, the implication of this incomplete biological cycle changes our entire framework for assessing the viability of extraterrestrial civilizations. The buried, unrotted wood of the Carboniferous eventually underwent millions of years of geological compression, transforming into the vast, subterranean coal seams that fueled the human industrial revolution. If we apply this to the Fermi Paradox, it reveals a terrifying temporal bottleneck. If an alien biosphere evolves lignin-digesting fungi just a few million years earlier than Earth did, dead plant matter would decompose immediately. The planet would never stockpile the easily accessible, high-density fossil fuels required to bootstrap a species from an agrarian society into a spacefaring one. A civilization could possess extreme intelligence, yet remain permanently trapped in a low-energy paradigm, unable to escape their planet's gravity, well, simply because their world's evolutionary timing was slightly too efficient. Furthermore, this unrepeatable geological window exposes the acute fragility of our own technological existence. Earth functions as a single-use biological battery. Because white rot fungi and termites now actively dismantle dead wood, the planet can never again generate coal on a Carboniferous scale. If our current technological infrastructure collapses, the easily accessible energy reserves required for a second industrial revolution no longer exist. Within this hyperoxygenated atmosphere, the physical limits of terrestrial biology were fundamentally rewritten. We witness the sudden, explosive gigantism of the arthropods, because insects and arachnids do not possess active, pump-driven lungs. They rely on a passive network of tracheal tubes, called spiracles, to deliver oxygen directly to their tissues. This system is normally severely limited by the mathematics of gas diffusion, acting as a strict ceiling on their physical size. But in an atmosphere composed of 35% oxygen, that mathematical limit was momentarily shattered. The fossil record yields the remains of Meganeura, a predatory griffinfly with a wingspan approaching 2.5 feet, dominating the aerial ecosystem. On the forest floor, Arthropleura, a heavily armored, multi-segmented millipede, reached lengths of nearly 8 feet, navigating the dense underbrush, devoid of large, vertebrate predators. But the issue does not stop at mere opportunity. For decades, the dominant scientific consensus was that high atmospheric oxygen simply permitted this massive growth. However, modern physiological debates introduce a much more severe mechanism: oxygen toxicity. At 35% concentration, oxygen is a highly reactive, volatile element that causes severe oxidative stress and cellular damage, particularly to the delicate respiratory membranes of developing larvae. A compelling competing hypothesis suggests that arthropod gigantism was not an opportunistic expansion, but a desperate physiological defense mechanism. By drastically increasing their physical volume, these organisms decrease their surface area to volume ratio, intentionally restricting their relative oxygen intake. They grew into monsters simply to prevent their internal tissues from chemically burning from the inside out. If this hypothesis is correct, it introduces a deep paradox into our understanding of habitable zones. The very chemical element that powers complex life was acting simultaneously as environmental fuel and an active environmental toxin, forcing extreme, high-stress morphological adaptations. The Carboniferous rainforests had engineered an atmosphere of unparalleled energetic potential, but it was an inherently unstable architecture, turning the surface of the Earth into a highly pressurized, highly flammable tinderbox that was exceptionally vulnerable to the slightest climatic shift. The hyper-oxygenated atmosphere of the Carboniferous was not a sign of a stable biosphere, but the symptom of a deeply broken planetary carbon cycle. For tens of millions of years, the Earth was systematically burying its own biological output. The sprawling equatorial forests were manufacturing millions of tons of lignin, the dense structural polymer that constitutes wood. However, biological evolution is not proactive. It does not invent solutions simply because a resource is abundant. The specific enzymatic keys required to dismantle the complex, randomized chemical bonds of lignin, specifically the specialized peroxidases utilized by modern white rot fungi, did not yet exist in the biosphere. This evolutionary blind spot created a catastrophic delay. Because the wood could not rot, the carbon locked within it could not return to the atmosphere as carbon dioxide. Dead forests collapsed into stagnant anoxic swamps, stacking hundreds of meters of pristine, undecayed, organic matter that was slowly crushed by geological pressure into the massive coal seams we mine today. But this continuous planetary-scale carbon sequestration inevitably triggered a severe climatic backlash. By aggressively pulling carbon dioxide out of the air to build their trunks, and failing to release it back through decomposition, the forests actively stripped the Earth of its thermal greenhouse blanket. The planet began to freeze. Massive glaciers expanded across the southern supercontinent of Gondwana, dropping global sea levels and fundamentally altering planetary weather patterns. The warm, humid equatorial belt shattered. The vast, continuous rainforest dried out, fragmenting into isolated patches, surrounded by expanding, arid deserts. This event, known as the Carboniferous rainforest collapse, suddenly rendered the dominant survival strategies of the era obsolete. The apex predators of these ancient swamps were colossal amphibians, some resembling modern crocodiles in size and ferocity. Yet their dominance was an illusion, permanently tethered to the water by a critical biological flaw. Amphibian reproduction relies on permeable, gelatinous eggs that require a continuous aquatic environment to prevent desiccation. As the swamps evaporated, the amphibian empire collapsed, entirely incapable of migrating across the newly formed dry continental interiors. And right here, this ecological crisis forced the invention of one of the most consequential biological technologies in the history of the Earth. From the marginalized, drying fringes of the collapsing rainforests emerged a lineage of tetrapods that had engineered a radical solution: the amniotic egg. This structure is, in essence, a microscopic, biologically sealed spacecraft. Instead of relying on a pond to protect the embryo, the amniotic egg packages the pond inside a semi-permeable membrane. It features a hardened outer shell that allows essential gas exchange while aggressively retaining moisture. Inside, it houses a dedicated nutritional payload in the yolk, and a highly specialized sac called the allantois to safely sequester toxic metabolic waste away from the developing organism. At a planetary scale, the meaning of this anatomical leap changes entirely. Before the amniotic egg, the continents were merely a temporary feeding ground. The deep, fundamental mechanism of reproduction still belonged exclusively to the ocean and the swamp. The amniotic egg permanently severed that evolutionary umbilical cord. It allowed vertebrate life to completely detach from the hydrological cycle of the coastlines and penetrate the deep, arid interiors of the supercontinents. If this precise reproductive containment architecture is not achieved, the evolution of a terrestrial, tool-using civilization is physically impossible. When we analyze the Great Filter and the probability of intelligent life across the cosmos, the transition from aquatic to fully terrestrial existence represents a massive bottleneck. An intelligent species might evolve in an alien ocean, but the foundational technologies of advanced civilization, the controlled combustion of fire, the smelting of heavy metals, and the generation of electricity cannot be developed underwater. They strictly require a dry atmospheric environment. The amniotic egg was the mandatory biological bridge that permitted a fragile, water-based nervous system to survive, expand, and eventually build a technological infrastructure on the dry, irradiated surface of a rocky world. The reproductive revolution of the amniotic egg allowed early tetrapods to survive the collapse of the Carboniferous swamps, but the subsequent Permian period introduced a geological filter of an entirely different magnitude. Approximately 299 million years ago, the slow, relentless engine of plate tectonics drove the Earth's major landmasses into a single, colossal collision. This formed the supercontinent Pangaea. From a planetary mechanics perspective, aggregating nearly all terrestrial surface area into one hemisphere fundamentally breaks the global hydrological cycle. The interior of Pangaea was so incredibly vast that moisture evaporated from the Panthalassic Ocean simply could not carry far enough inland before precipitating. The center of the supercontinent became a permanent, hyper-arid wasteland, locked in a state of severe hydrological starvation. At a planetary scale, the meaning of this geographical configuration alters our parameters for cosmic habitability. When astronomers search for habitable exoplanets, a stable climate is often prioritized. But Pangaea demonstrates that absolute stability is the enemy of radical biological innovation. A world composed entirely of small islands or scattered archipelagos might foster a comfortable, unbroken ecosystem, but it lacks the extreme, inescapable environmental stress required to force a paradigm shift. Supercontinents function as planetary pressure cookers. If an alien world lacks the active tectonic machinery to periodically assemble and shatter supercontinents, its biosphere might remain permanently trapped in a state of low-level, comfortable stagnation. The deep interior of Pangaea was not merely dry, it was thermodynamically violent. Without the temperature-regulating presence of atmospheric water vapor, the landmass experienced brutal diurnal temperature swings, baking under lethal solar radiation during the day and plunging below freezing at night. For the dominant reptiles of the early Permian, this presented an insurmountable mechanical limit. As ectotherms, their internal body temperature was entirely dictated by the ambient environment. During the freezing nights, their metabolic engines effectively shut down, leaving them paralyzed and vulnerable until the morning sun could re-energize their tissues. We see the first desperate evolutionary attempts to solve this thermodynamic problem in the apex predators of the era, the pelycosaurs. Creatures like Dimetrodon evolved massive, highly vascularized dorsal sails supported by elongated neural spines. For decades, these structures were debated, but the prevailing biomechanical model suggests they functioned as active heat exchangers. By turning broadside to the early morning sun, a Dimetrodon could rapidly pump cold blood through the expansive surface area of the sail, artificially raising its core temperature hours before its competitors could even move. But the problem does not stop at raw heat collection. A passive solar array is a geographic and meteorological tether. If the sky is overcast, or if the animal needs to hunt in the deep shade, the biological machinery stalls. Passive thermoregulation represents an evolutionary dead end for complex behavior. And right here, this physical limitation forces one of the most consequential anatomical transitions in the history of life. From the lineage of the pelycosaurs emerged the therapsids, the direct stem ancestors of all modern mammals. To conquer the relentless thermal chaos of the Pangaean interior, the therapsids abandoned the external sail and engineered an internal furnace. This is the dawn of endothermy. Fossil evidence from early therapsids reveals the restructuring of the nasal cavity to accommodate respiratory turbinates, complex bony scrolls designed to warm and humidify inhaled air while recapturing moisture during exhalation. This microscopic architectural shift proves that these organisms were breathing at an intensely elevated rate, fueling a constant, high-output metabolic engine that generated its own internal heat. This leads to a much deeper consequence in the biological timeline. Endothermy is an agonizingly expensive biological strategy. A warm-blooded organism requires up to 10 times the caloric intake of a cold-blooded counterpart of the same size. It is a biological gamble that demands relentless hunting and foraging simply to keep the internal fires burning. However, the payoff is absolute ecological liberation. A therapsid was no longer a slave to the position of the sun. It could hunt in the dead of night, migrate across freezing latitudes, and sustain prolonged high-speed muscular exertion without rapidly building up paralyzing lactic acid. If this thermodynamic leap had not occurred, the trajectory toward technological civilization would have been physically blocked. When we consider the Fermi Paradox, the development of endothermy acts as a silent, invisible, great filter. High-level intelligence requires a massively centralized nervous system. The mammalian neocortex is an incredibly fragile biological computer that demands a strictly controlled, unvarying thermal environment to process data efficiently. A temperature drop of just a few degrees causes catastrophic neurological failure. Without the brutal, hyper-arid crucible of Pangaea forcing our primitive ancestors to internalize their climate control, the stable neuroenvironment required to eventually map the genome, split the atom, and comprehend the expansion of the universe could never have been sustained. The biological innovations of the early Permian were a triumph of adaptation, but they were fundamentally blind to the geological time bomb ticking deep within the mantle. 252 million years ago, the stability of the Pangaean supercontinent was shattered by an event of such absolute devastation that it nearly reset the planetary evolutionary clock to zero. This is the Permian-Triassic extinction event, often referred to as the Great Dying. The fossil record indicates a staggering loss. Over 95% of marine species and more than 70% of terrestrial vertebrate life completely vanished. Yet, the true weight of this event lies not in the final death toll, but in the intricate, cascading failure of the entire planetary operating system. The trigger was not falling from the sky, it was rising from the core. Beneath the region that is now Siberia, a colossal mantle plume, a superheated upwelling of magma, burned its way through the continental crust. This did not create a single mountain, but a massive tear in the earth, unleashing a flood basalt eruption known as the Siberian Traps. For hundreds of thousands of years, lava flowed endlessly, burying an area roughly the size of Western Europe under kilometers of newly forged rock. But the raw volume of magma was merely the physical catalyst. The true weapon of mass extinction was chemical, and it was deeply tied to the Earth's biological past. As the magma pushed upward, it did not just melt rock. It intruded directly into the massive subterranean coal basins that had been laid down during the Carboniferous period millions of years prior. The Earth essentially ignited its own fossil fuel reserves. The extreme geological heat vaporized gigatons of coal, oil, and gas, violently ejecting unprecedented volumes of carbon dioxide and methane directly into the atmosphere. This leads to a systemic consequence that redefines our understanding of planetary boundaries. The sudden, extreme spike in greenhouse gases triggered hyperwarming on a global scale. Equatorial ocean surface temperatures soared to lethal levels, approaching 104 degrees Fahrenheit. Because warm water physically cannot hold as much dissolved oxygen as cold water, the ocean's global conveyor belt of thermohaline circulation abruptly stalled. The deep ocean stagnated, creating an immense planet-wide dead zone. The anoxic catastrophe of the Devonian had returned, but this time, the scale was absolute. And right here, this geochemical collapse initiates a far more sinister biological mechanism. As the oxygen-breathing life died and sank into the suffocating depths, a completely different, ancient empire reclaimed the oceans. In the total absence of oxygen, anaerobic microbes known as sulfate-reducing bacteria experienced an explosive global population bloom. These primitive organisms rely on a different metabolic pathway, stripping oxygen from dissolved sulfate molecules and releasing a highly toxic byproduct, hydrogen sulfide gas. The sheer scale of this microbial bloom fundamentally transformed the chemistry of the global ocean. The water column saturated with hydrogen sulfide, eventually turning the sunlit surface waters a toxic, bruised purple. But the problem did not stop at the water's edge. As the ocean's carrying capacity for the gas was exceeded, massive bubbles of lethal hydrogen sulfide erupted into the atmosphere, drifting over the coastlines and sweeping across the continental interior. At a planetary scale, the implication of this mechanism changes entirely how we must search for life in the universe. If we point our instruments at a distant exoplanet and detect high concentrations of hydrogen sulfide in its atmospheric spectrum, we might not be looking at a young pre-oxygen world. We could be witnessing the terminal phase of a collapsing biosphere. The Permian-Triassic extinction provides a terrifying localized model for the Great Filter. It demonstrates that the greatest threat to advanced life is not necessarily astronomical, but the inherent, lethal feedback loops written into the chemistry of an active planet. A world can foster highly evolved ecosystems, only to suffocate them under a blanket of toxic gas generated by the simplest microscopic organisms reacting to a temporary geological anomaly. The atmospheric cascade continued upward. As hydrogen sulfide gas reached the stratosphere, it reacted violently with the ozone layer, systematically dissolving the planet's ultraviolet shield. The terrestrial ecosystems, already baking under the extreme greenhouse heat, were suddenly bombarded by sterilizing cosmic radiation. We see the direct, irrefutable evidence of this in the fossil record, where surviving pollen grains and plant spores from the late Permian exhibit severe radiation-induced genetic mutations. The foundational base of the terrestrial food chain simply disintegrated. The flora died, the herbivores starved, and the apex therapsid predators, despite their advanced endothermic biology, followed them into oblivion. If this chain reaction had been even fractionally more efficient, Earth would have been sterilized down to the microbial level, permanently aborting the trajectory toward the dinosaurs, the mammals, and eventually human intelligence. The Great Dying forces us to acknowledge a profound and uncomfortable reality regarding our own technological civilization. We are currently extracting and burning the exact same ancient carbon reservoirs that the Siberian traps ignited, artificially mimicking the primary trigger of the most devastating mass extinction in planetary history. The ultimate lesson of the Permian is that the Earth's atmospheric chemistry is not a permanent sanctuary. It is a highly volatile, strictly balanced equation. Push the thermal limits of the oceans too far, and the planet possesses deeply buried biological fail-safes that will automatically and systematically dismantle the higher tiers of the food web. The aftermath of the Permian-Triassic extinction presents a geological landscape that resembles a sterilized alien world. Entering the early Triassic period, roughly 252 million years ago, the Earth did not experience a rapid biological rebound. Instead, the planet remained locked in a state of severe ecological trauma for millions of years. The fossil record from this epoch reveals terrifying planetary anomalies, a complete absence of coral reefs in the oceans, and a massive, global coal gap on land, indicating that the terrestrial forests had been so thoroughly annihilated that the planet temporarily lost the ability to generate massive, organic deposits. The supercontinent Pangaea was a vast, baked superdesert, swept by mega monsoons that triggered catastrophic flash floods across a landscape largely devoid of root systems to hold the soil together. But the most critical challenge for surviving terrestrial life was invisible. The violent volcanic outgassing that ended the Permian had fundamentally altered the atmospheric ratio. Global oxygen levels, which had peaked at 35% during the Carboniferous, plummeted to an estimated 12 to 15%. To understand the severity of this shift, breathing at sea level in the early Triassic would be the physiological equivalent of standing near the summit of the modern Himalayas. This introduces a severe biomechanical filter. The dominant survivors of the extinction were the therapsids, specifically a pig-sized burrowing herbivore known as Lystrosaurus, which temporarily constituted over 90% of all terrestrial vertebrates. They survived the thermal radiation of the surface by retreating underground. However, their internal architecture contained a fatal flaw inherited from their amphibian ancestors. Therapsids, and modern mammals by extension, utilize a tidal respiratory system. We draw air into a dead-end sac, the alveoli, extract the oxygen, and push the stale air back out the same pathway. This means fresh air and depleted carbon dioxide continuously mix in the lungs. In a high oxygen environment, this inefficiency is manageable. In a hypoxic, Triassic wasteland, it is a metabolic death sentence. A therapsid, attempting sustained physical exertion in this thin atmosphere, would rapidly collapse from oxygen starvation and lactic acid buildup. And right here, this atmospheric bottleneck sets the stage for a biological coup that would dictate the next 160 million years. From the margins of the devastated ecosystems emerged a new lineage of reptiles, the archosaurs, the ruling reptiles, whose descendants include modern crocodiles and birds. To survive the low oxygen conditions, early archosaurs evolved a radically different fluid dynamics model for respiration. They abandoned the dead-end lung. Instead, they developed a system of unidirectional airflow. By utilizing a complex network of flexible air sacs distributed throughout their bodies, they separated the mechanical action of pumping air from the chemical action of gas exchange. When an archosaur inhaled, air moved into rear sacs. When it exhaled, that fresh air was pushed across rigid lungs in a single, continuous direction, while stale air was simultaneously forced out. This allowed for countercurrent gas exchange, an engineering marvel where blood flows in the opposite direction of the continuous airstream, extracting nearly every available molecule of oxygen with brutal efficiency. At a planetary scale, the implication of this respiratory arms race fundamentally alters how we view the evolution of complex biospheres. The dominance of a species is not necessarily dictated by intelligence or raw strength, but by microscopic chemical efficiency. If an alien world orbiting a red dwarf star maintains a permanently low oxygen atmosphere, its apex predators will undoubtedly utilize unidirectional fluid mechanics rather than mammalian tidal breathing. The biological constraints of a planet's atmosphere dictate the very blueprints of its dominant life forms. Armed with this hyper-efficient respiratory engine, the archosaurs were able to sustain high-speed pursuit, outmaneuver, and outlast the sluggish therapsids. They rapidly expanded, radiating into a staggering array of apex predators, armored herbivores, and eventually the first true dinosaurs. This leads to a much deeper consequence for our own evolutionary timeline. The rise of the archosaurs did not simply defeat our therapsid ancestors, it systematically crushed them. The surviving stem mammals were driven out of the daylight entirely. To avoid being hunted to extinction by highly active, visually acute archosaur predators, our ancestors were forced to adapt to the deep night. They shrank in size, abandoning the massive, energy-intensive bodies of the Permian. But this absolute defeat is arguably the most crucial turning point in the history of human intelligence. Survival in the dark required thermal insulation, leading to the evolution of mammalian fur. Nighttime navigation rendered vision secondary, forcing a massive expansion of the olfactory and auditory processing centers in the brain. The necessity of keeping offspring safe from nocturnal predators drove the internalization of gestation and the production of milk. If this is true, it connects directly to the Great Filter and the sheer statistical improbability of an advanced civilization. The hyper-advanced, centralized brain required to eventually comprehend mathematics and build radio telescopes was not forged in an era of dominance. It was an indirect byproduct of severe ecological suppression. If the atmospheric oxygen levels had not crashed in the Triassic, if the archosaurs had not evolved their superior lungs, the therapsids might have remained large, diurnal herbivores, and the specific, intense pressures required to forge the complex mammalian neocortex might never have existed. The path to technological intelligence required our ancestors to lose the world, spending 100 million years hiding in the shadows of giants. As the Triassic gave way to the Jurassic period roughly 201 million years ago, the supercontinent of Pangaea began to fracture. The violent tectonic rifting opened new ocean basins, pumping immense quantities of water vapor into the atmosphere and transforming the arid superdesert into a lush, global greenhouse. Within this hyper-productive botanical landscape, the respiratory advantage of the archosaur lineage was pushed to its absolute physical extreme. We witnessed the rise of the sauropods, the colossal, long-necked dinosaurs that stand as the largest terrestrial animals in planetary history. For over a century, biomechanical models struggled to explain their existence. According to the rigid mathematics of the square-cube law, an animal scaling up to 70 or 80 tons should physically collapse under its own weight, its solid bones shattering from the sheer gravitational stress of movement. The solution to this paradox was not found in the strength of their bones, but in their emptiness. Modern high-resolution CT scans of sauropod fossils reveal a skeletal architecture that is fundamentally alien to the mammalian blueprint. Their massive vertebrae were not solid blocks of calcium. They were extensively hollowed out, featuring a complex honeycomb structure composed of impossibly thin bony struts. This phenomenon, known as postcranial skeletal pneumaticity, indicates that the unidirectional air sac system, originally evolved to survive the hypoxic Triassic wasteland, had literally invaded the skeleton. By replacing heavy marrow with air, a sauropod could expand its physical volume without a corresponding linear increase in mass. At a planetary scale, the implication of this skeletal pneumatization completely alters our biological models for exoplanetary habitability. When astrophysicists model super-Earths, rocky planets with 2 or 3 times the gravitational pull of our world, it is often assumed that any terrestrial life would be flat, sprawling, and heavily restricted in size. However, the sauropod blueprint suggests an alternative. If alien evolution discovers the mechanism of internal pneumatization, biological organisms could effectively hack the local gravity well, utilizing gas-filled structural voids to achieve macroscopic size even under crushing gravitational conditions. But this immense scale introduces a severe, localized mechanical crisis: the hemodynamics of the long neck. To operate a brain suspended 10 to 12 meters above the heart requires generating a systemic blood pressure so extreme that it defies the limits of mammalian tissue. If a giraffe's relatively modest blood pressure requires a specialized, hypermuscular heart, pumping fluid to the altitude of a brachiosaurus head would theoretically generate enough hydrostatic pressure to rupture the capillaries in the brain or cause catastrophic blood pooling in the lower limbs. This leads to a deeply contested debate within modern paleontology regarding the limits of biological engineering. Some researchers propose the existence of multiple auxiliary biological pumps distributed along the neck, a hypothesis lacking fossil evidence but demanded by fluid dynamics. Others suggest a simpler behavioral constraint. Sauropods rarely lifted their heads vertically, instead utilizing their necks as horizontal sweepers to clear vast arcs of low-lying vegetation without moving their massive, energy-intensive bodies. This debate highlights a critical threshold in biology. There is a definitive point where the physical laws of hydrodynamics permanently veto further evolutionary expansion, acting as a strict, localized ceiling on planetary life forms. Furthermore, the unidirectional air sac system solved a second, equally lethal problem of gigantism: thermodynamics. A 70-ton endotherm generates a catastrophic amount of internal metabolic heat without sweat glands and possessing a highly unfavorable surface area to volume ratio. A sauropod should theoretically cook from the inside out. The pervasive network of air sacs extending throughout their core acted as a highly efficient internal radiator, constantly venting waste heat directly from deep tissue into the exhaled airstream. The respiratory system that saved their ancestors from suffocation had been repurposed into an industrial-scale biological cooling tower. And right here, this exact architectural blueprint connects to a radically different evolutionary trajectory, unfolding at the opposite end of the size spectrum. While the sauropods leveraged the air sac system to achieve maximum terrestrial volume, a lineage of small, hyperactive, carnivorous theropods utilized the identical respiratory engine to push their metabolic rates to extreme limits. But a tiny endothermic body loses heat rapidly to the environment. To survive, these small theropods required intense thermal insulation, driving the widespread evolution of complex filamentous structures covering their skin, the first feathers. This brings us to a profound biomechanical convergence preserved in the limestone quarries of the late Jurassic. The discovery of Archaeopteryx reveals a creature possessing the teeth and long bony tail of a theropod, yet covered in fully developed asymmetric flight feathers. The emergence of avian flight was not a conscious evolutionary goal. It was the accidental synthesis of pre-existing survival mechanisms. The hollow pneumatic skeleton evolved to reduce mass. The unidirectional lungs evolved to extract oxygen efficiently, and the feathers evolved to trap heat. When combined in a small muscular frame, these three independent variables unexpectedly unlocked the physics of aerodynamic lift. If we examine this transition through the lens of the Fermi Paradox, The escape from the two-dimensional terrestrial plane represents a monumental cognitive and physical leap. Mastering the three-dimensional fluid matrix of the atmosphere forces a radical upgrade in neurological processing, specifically in spatial mapping, balance, and real-time trajectory calculation. Biological flight is the planetary prototype for understanding fluid dynamics and aerodynamics. If a biosphere fails to produce this specific convergence of lightweight architecture and high-energy metabolism, its inhabitants remain bound to the ground. They might develop complex societies, but without the innate biological understanding of the sky. The subsequent conceptual leap to artificial aviation, orbital mechanics, and eventual spaceflight becomes a significantly steeper, perhaps insurmountable, intellectual barrier. The complete disintegration of the supercontinent during the Cretaceous period, beginning roughly 145 million years ago, physically dismantled the global biological monoculture. As massive tectonic rifts tore the landmasses apart, the Atlantic Ocean opened, and extreme global warming melted the polar ice, raising sea levels to unprecedented heights. Vast epicontinental seas flooded the continental interiors, slicing major landmasses into heavily isolated archipelagos, such as the island continent of Laramidia in what is now Western North America. This geographic fragmentation initiated the most extensive, high-speed experiment in allopatric speciation in planetary history. Evolution was no longer operating on a single, continuous data set. Instead, the Earth became a matrix of dozens of isolated biological laboratories. Without gene flow between these separate populations, localized environmental pressures forced extreme physiological specialization. We see the emergence of highly distinct regional dinosaur faunas, from the massive horned ceratopsians of the north to the specialized abelosaurid predators of the southern hemisphere. At a planetary scale, the meaning of this tectonic isolation fundamentally alters our parameters for evaluating long-term biological resilience. In the search for extraterrestrial life, a planet with a monolithic, unbroken landmass presents a critical vulnerability. A single, globally connected ecosystem is highly susceptible to localized diseases or sudden climatic shifts cascading across the entire system. Tectonic fragmentation acts as a planetary defense mechanism, artificially compartmentalizing the biosphere. It creates independent biological backups. If an alien world lacks the active geological machinery to continuously divide and isolate its ecosystems, its evolutionary trajectory might remain permanently fragile, highly vulnerable to total extinction from a single ecological collapse. But the most consequential development of the Cretaceous was not the geographical isolation of the dinosaurs. It was a microscopic revolution occurring at the very base of the food web. For hundreds of millions of years, terrestrial flora, the ferns, cycads, and massive conifers, relied entirely on the wind to distribute their pollen. This is a blind, passive, and staggeringly inefficient reproductive strategy, requiring the plant to manufacture billions of pollen grains simply to guarantee a few successful microscopic collisions. Deep within the early Cretaceous strata, we find the solution to this mathematical inefficiency: the sudden, explosive radiation of the angiosperms, the flowering plants. To bypass the randomness of the wind, the angiosperms engineered a targeted, active transport system. They outsourced their reproductive logistics to the highly mobile insect populations. The flower itself is not merely a botanical display. It is a highly advanced, multi-sensory optical and chemical beacon. By generating vivid ultraviolet patterns invisible to vertebrate eyes and synthesizing complex aromatic compounds, the plant broadcasts a specific electromagnetic and chemical frequency to attract a precise pollinator. To guarantee compliance, the flower manufactures nectar, a high-density, easily synthesized sugar compound that serves absolutely no internal biological function for the plant itself. It is pure biological currency. This establishes a profound co-evolutionary contract. The insect receives immediate, high-octane metabolic fuel, and the plant achieves highly accurate, long-distance, genetic transfer. This leads to a systemic consequence that fundamentally upgraded the planetary energy budget. Because angiosperms no longer relied on brute force pollen production or specific wind conditions, their reproductive cycles became vastly shorter and more resilient. They could rapidly colonize devastated landscapes, outcompeting the slow-growing conifers. More importantly, to protect and distribute their fertilized seeds, angiosperms encase them in energy-dense, specialized tissues, the first true fruits. And right here, this botanical mechanism begins to connect with the ultimate timeline of human civilization. The co-evolution of flowers and pollinating insects, such as early bees and butterflies, injected an unfathomable caloric surplus into the terrestrial ecosystem. The global proliferation of fruit, nectar, and complex, easily digestible seeds provided a massive, reliable energy baseline that had never previously existed. If we examine this through the lens of the Great Filter and the prerequisites for technological intelligence, the angiosperm revolution is an absolute, non-negotiable threshold. The immense metabolic demands of the mammalian endothermic engine, and specifically the hyper-expensive energy requirements of the primate neocortex, cannot be sustained by grazing on tough, low-calorie pine needles or ferns. Furthermore, the very foundation of human civilization, agriculture, is almost entirely reliant on angiosperms. The cultivation of wheat, rice, corn, and fruit-bearing trees is nothing more than the industrial hijacking of this specific Cretaceous technology. If the terrestrial biosphere had failed to invent the flower, the caloric density required to transition a species from nomadic hunting to static, city-building agriculture would simply not exist. An intelligent alien species evolving on a world dominated solely by wind-pollinated flora might possess the neurological capacity for civilization, but they would be permanently starved of the baseline agricultural fuel necessary to bootstrap an industrial revolution. The architecture of our modern world was ultimately designed by the intricate, fragile negotiations between primitive insects and the first petals opening under the Cretaceous sun. 66 million years ago, the highly optimized, hyper-specialized biosphere of the late Cretaceous was abruptly dismantled by a mathematical absolute, orbital mechanics. A piece of carbonaceous chondrite, roughly 10 kilometers in diameter, intersected Earth's orbit. The sheer size of the Chicxulub impactor is often cited as the primary cause of the extinction, but modern geophysical modeling reveals a much more terrifying reality. The lethality of the event was not dictated merely by mass, but by the exact physics of the impact vector and the specific geological chemistry of the target. When the asteroid struck the shallow sea of what is now the Yucatan Peninsula, it did not hit straight down. Core samples drilled directly from the peak ring of the impact crater indicate that the asteroid arrived at an angle of roughly 60 degrees. From a ballistic physics perspective, this is the deadliest possible trajectory. A perfectly vertical strike would have driven the kinetic energy deep into the mantle, containing much of the blast beneath the crust. A shallow grazing impact would have deflected a massive portion of the energy back into space. But a 60-degree strike acts like a planetary-scale shotgun blast. It maximizes the volume of crustal rock that is pulverized, instantly melted, and ejected upward. This introduces a critical parameter into our understanding of planetary defense and civilizational longevity. When we look outward and model the survivability of intelligent life on other worlds, it becomes clear that surviving a cosmic impact is not just about the size of the rock. It is an agonizing game of geological roulette. The exact spot the asteroid hit was one of the most volatile regions on the planet, a massive shelf of anhydrite and gypsum. The kinetic energy of the strike vaporized this rock instantly, injecting billions of tons of vaporized sulfur directly into the upper atmosphere. If the Earth had been rotating just 30 minutes faster or slower, the asteroid would have plunged into the deep Pacific Ocean. It would have triggered colossal tsunamis, but it would not have vaporized the sulfur shelf in nearly the same volume. The catastrophic global climate shift would not have occurred to the same extreme, and the dinosaurs would likely have survived. Human intelligence exists solely because of a 30-minute margin of error in a cosmic timeline spanning billions of years. But the immediate devastation at the impact site was only a localized phenomenon. As the 60-degree strike violently evacuated the crater, it launched trillions of tons of superheated plasma and vaporized rock completely outside the Earth's atmosphere. This ejector plume followed a suborbital ballistic trajectory, briefly entering the vacuum of space before condensing into tiny, liquid glass spheres known as tektites. And right here, this physical mechanism initiates a global thermal shockwave that defies conventional comprehension. As these trillions of glass spherules succumb to gravity, they re-enter the atmosphere simultaneously across the entire planet. The sheer friction of this global micrometeorite shower heated the upper atmosphere to incandescent temperatures. For a few agonizing hours, the sky itself turned into a massive red-hot radiator. The thermal pulse delivered to the surface is estimated to have been over 10 kilowatts per square meter, essentially turning the entire surface of the Earth into a broiler oven. This radiant heat was intense enough to spontaneously ignite exposed biological matter. Vast swaths of the Cretaceous forests simultaneously burst into flames across the Americas, Eurasia, and Africa, generating a global firestorm. The only organisms capable of surviving this immediate thermal flash were those shielded by thermal mass. Amphibians deep underwater, small mammals hidden in subterranean burrows, and reptiles sheltering inside rotting logs. This leads to a profound realization about the Great Filter and the architecture of survival. Dominance on the surface of a planet is actually a liability during a cosmic intervention. The colossal sauropods and the apex theropod predators, despite their millions of years of rigorous evolutionary optimization, were completely defenseless against an attack from the upper atmosphere. Their massive bodies, which provided absolute security against biological threats, became inescapable thermal traps. The architecture of the Earth's biosphere was instantly inverted. The marginalized, the small, and the hidden, those lineages that had been aggressively suppressed by the Archosaur dynasty for over 100 million years, were suddenly the only biological entities equipped with the exact behavioral parameters required to survive a global atmospheric ignition. The global firestorms that ravaged the Earth's surface were merely the opening phase of a much longer, darker climatic nightmare. As the fires finally exhausted the available oxygen and fuel, the planet did not return to its lush Cretaceous baseline. Instead, the billions of tons of vaporized sulfur and pulverized bedrock ejected by the Chicxulub impact, combined with the immense volume of soot from the burning forests, accumulated in the stratosphere. This created an impenetrable aerosol shield. For months, perhaps years, the Earth was plunged into near-total darkness. This initiates the impact winter, a physical mechanism that fundamentally breaks the primary biological engine of the planet: photosynthesis. Without incoming solar radiation, the microscopic phytoplankton in the oceans and the surviving vegetation on land ceased to produce energy. The collapse was immediate and mathematically absolute. The giant herbivorous dinosaurs, requiring hundreds of pounds of fresh vegetation daily to fuel their massive endothermic engines, starved within weeks. The apex predators, finding only rotting carcasses, soon followed. But the problem extended deeply into the oceans. As the atmospheric sulfur dioxide mixed with atmospheric moisture, it rained down as highly concentrated sulfuric acid, drastically lowering the pH of the surface waters. This rapid ocean acidification dissolved the calcium carbonate shells of the calcareous plankton, causing the complete collapse of the marine food web and eradicating the giant marine reptiles that had dominated for eras. At a planetary scale, the meaning of this atmospheric occlusion completely changes our understanding of biological resilience. When astrobiologists consider the longevity of an alien biosphere, the ultimate vulnerability is its reliance on a host star. An ecosystem optimized entirely for sunlight is inherently fragile. The Chicxulub event demonstrates that a biosphere can only survive a cosmic intervention if it possesses a robust, secondary shadow ecology, a network of organisms that do not rely on active primary production, but instead feed on the decaying remnants of the system itself. And right here, this grim ecological reality perfectly aligns with the survival of our direct ancestors. The early mammals did not endure the impact winter because they were stronger, smarter, or genetically superior in a traditional sense. They survived because they were biologically pre-adapted to exist in a dead world. For 100 million years, the terrifying dominance of the archosaurs had forced mammals to become nocturnal, burrowing creatures. This evolutionary suppression inadvertently engineered the exact physiological profile required to survive an apocalypse. When the thermal flash incinerated the surface, mammals were already safely insulated deep underground in complex burrow systems. When the global temperature plummeted during the impact winter, their endothermic metabolism, combined with the insulating layer of fur they had evolved for the Cretaceous nights, prevented them from freezing to death. But the most critical survival mechanism was their diet. The mammals of the late Cretaceous were largely insectivores and scavengers. While the giant herbivores starved in the dark, the dead forest provided an almost limitless supply of rotting wood and decaying organic matter. This fueled an explosive, temporary bloom of fungi and detritus-eating insects. The mammals simply shifted their foraging strategies to exploit this subterranean, rot-based food web. They survived by eating the creatures that were eating the ruins of the old world. If this specific sequence of ecological suppression and opportunistic scavenging had not occurred, the evolutionary lineage leading to human civilization would have been completely eradicated. This introduces a profound paradox into the Great Filter hypothesis. We often assume that the species destined to build a technological civilization will naturally be the dominant, apex organisms of their respective worlds. But the geological record dictates the exact opposite. True planetary dominance is a vulnerability. It creates a highly specialized biological rigidity that shatters under sudden cosmic stress. The traits necessary to weather a planetary cataclysm, small size, extreme dietary flexibility, and the ability to hide from the dominant hierarchy, are forged only in the margins. The ultimate lesson of the impact winter is that the universe systematically prunes the majestic and the highly optimized, leaving the future of the planet entirely to the scavengers waiting in the dark. When the atmospheric dust of the Chicxulub impact finally settled, it revealed a planet completely stripped of its biological incumbents. For over 100 million years, the Archosaur lineage had monopolized the sunlit terrestrial niches, exerting a crushing evolutionary pressure that locked mammalian life into a permanent subterranean holding pattern. The mass extinction did not destroy the biosphere, it defragmented it. In evolutionary biology, this is recognized as the violent removal of the incumbent effect. By abruptly deleting the highly optimized apex organisms, the asteroid artificially engineered an ecological vacuum, suddenly opening up vast, empty trophic networks that had been physically inaccessible to mammals for eras. At a planetary scale, the meaning of this reset alters our entire equation for the Fermi Paradox and the search for extraterrestrial intelligence. We inherently view long-term planetary stability as the ultimate prerequisite for advanced life. Yet, the geological record insists on the exact opposite. A perfectly stable, peaceful world is a biological trap, without periodic catastrophic disruptions, whether triggered by deep mantle plumes, oceanic asphyxiation, or kinetic cosmic strikes. An ecosystem achieves a state of optimized stagnation. The ruling species become so perfectly adapted to their environment that they actively suppress all radical innovation. If an alien world never experiences its own planetary scale mass extinctions, it is highly probable that its biological timeline simply flatlines at the level of highly efficient, non-sentient apex predators. This leads to a profound realization about the emergence of our own technological existence. The dawn of the Cenozoic era, the age of mammals, was not a victory won through inherent genetic superiority, but an inheritance granted by random cosmic violence. The surviving mammals, previously restricted to extreme metabolic efficiency and nocturnal scavenging, suddenly found themselves in a sterilized world bathed in sunlight. This unprecedented ecological freedom triggered an explosive adaptive radiation. Within a few million years, these small burrowing survivors rapidly expanded in physical volume and neurological complexity, diversifying into the vast lineages of ungulates, cetaceans, and eventually the arboreal primates. We are not the culmination of a peaceful, linear evolutionary ascent. The specific neurological architecture required to construct cities, decode genomes, and build radio telescopes would have remained permanently suppressed in the dark if the old world had not been shattered. We are the direct, mathematically improbable descendants of the Earth's most devastating planetary resets.